This is going to be a little different from the talks that you've had up to now because I'm a neuroscientist, so that's entirely unlike any of the other talks. But I research space. I've been interested for a long time in how the brain builds this internal representation of space. And over the years, I've become increasingly interested in architecture and how that relates. And a lot of the reason is that my experience of architecture as someone who has some difficulties with space has been extremely frustrating and on an almost daily basis. Like today, for example, when I was trying to find my way around London. And knowing what we know now about how the brain organizes space, it seems to me that architects and cognitive neuroscientists should be having a lot more conversations than they seem to have. So I've managed to wangle my way into this program, and I really appreciate having this opportunity to tell you a little bit about some of the really big discoveries that have happened in the last few decades that have really transformed how we think about psychology and also given us a lot of insights into space. So architects, I love architecture, and architects are able to build some really beautiful spaces, and this is an example of what I think is quite a beautiful space. But more often than not, our spaces look like this. This is the building that I hate most in the world. It's the Washington, I hope none of you designed it. There's the Washington DC Convention Center where we have our annual neuroscience meeting. So 30,000 neuroscientists descend on this space that's about a mile long, and almost all of it looks like this, what you see in the picture. And if you turn around, you suddenly don't know which way you're facing, and you discover that you have to walk from one end of this building to the symposium, which is in 5 minutes at the other end of the building. And not only that, but it has a mirror symmetry across a road, so that if you've forgotten whether you were in the north or the south building, you could end up going east instead of west, and by the end of the day, you are completely exhausted. So it's not the only confusing building, there are others. Here's another very confusing building. It's very beautiful, I will admit it's beautiful, but it's very easy to just feel perplexed and end up following signs. This is possibly my favorite example because this has had an entire book written about how confusing it is by Ruth Dalton, who actually trained at the Bartlett. And it was... I think it won prizes. I mean, it's a very beautiful building, but it was so difficult to navigate that by the end of the first day, the librarians were having to put up signs saying, books this way, stairs that way, and so on. So it was an extremely difficult building to navigate. So my question is, can understanding something about the neuroscience of navigation help us with designing spaces that are less confusing? So I'm going to give you a very, very quick tour through the types of things that we do, and then I'll tell you a little bit about some of the things. It's just a small taster of some of the things that have been coming up. So there are too many methods to describe, but the broad ones, which have been used for a very long time and are in common use today, are still the same. So mostly we have two broad lines of inquiry. We study humans and we study their behavior and to some extent the neurobiology, but obviously we're limited in what we can do with humans. To really understand the neurobiology at a very fine-grained detail, we go and look at animals, and the reasoning is that a lot of these processes evolved a very long time ago, and rats and mice have them, and humans have them, and all sorts of other animals have them as well. So we're looking at very fundamental processes here. And everything that we find seems to suggest that that's true. So a lot of anatomical methods are used and have been for a very long time to just understand which parts of the brain are connected to which other parts. So these pictures here, so I haven't got a pointer, unfortunately, so I have to kind of wave at you. But these methods that were developed over 100 years ago now, the method of Golgi, which enabled us to dye neurons with a colored dye so that you can see them under a microscope, and the method of Cajal, who really, he was an artist as well as a scientist, and he mapped out the brain using Golgi's method and formed this very, very detailed wiring diagram of the brain. And then using more modern methods, we've managed to work out pretty much what's connected to what, and it seems that pretty much everything is connected to everything else. So it's quite a difficult problem to unravel, but we're on it. So the second really common method is to use single neuron recordings. So this is the method that we use in rats and mice most often. So The idea is that if you want to understand the language that the neurons are using to talk to each other, as it were, you need to be able to isolate what one of the neurons is saying and what its neighbors are saying and so on, and what messages are going backwards and forwards. So what you can see here is a slice through my favorite part of the brain, the hippocampus. And all the little colored dots are neurons, and they've been labeled with a genetic trick, if you like. So what they did was to make a mutant mouse whose genes encode not just the proteins for making neurons, but also the proteins for making a fluorescent dye, which lights up if you shine a fluorescent light on it. So these neurons kind of obediently make all of these proteins plus the fluorescent dye, and then anatomists can shine the light and light all the neurons in different colors and trace the pathways from the neurons to each other. So what the schematic rod shows you is the size of a typical microelectrode. So we will put these very, very fine wires, the thickness of a hair, into the brain. The brain has no pain-sensitive nerves in it, so this is a completely painless process. And in fact, human patients with epilepsy often have these electrodes implanted as well. And with these wires, we can detect the electrical impulses from the neurons and listen to the patterns and try and figure out the code, if you like. And I'll come back to that in a second. The third really common method, which it was developed in the 1990s, which we use mostly on humans, is to actually image the living, thinking brain using an extremely strong magnet. And what this does is it detects the blood flow through the brain and works out where the hot spots are. So if you're thinking in a particular way and using a particular part of the brain, then that part of the brain needs more blood supply to meet its oxygen needs. And that is picked up by our magnetic scanners, and they show up as hotspots. So what you can see on the bottom right there is a typical picture that would be generated by a subject who's, I'm not sure what they're doing, but they're probably navigating, actually. So with these hotspots, we can get some insights as to what parts of the brain people use when they're doing various things. And for example, using virtual reality, we can put people in the scanner and take them on a virtual tour through a landscape or through a building and find out what parts of the brain are they using. And then if they're given a task, like find your way from this building in this street to that building and that street, what part of the brain lights up. So we've got some amazing methods to look at the brain and what's going on there. So over the decades since we've been doing this, we've discovered that the brain has several different systems for processing space, not just one. For example, the type of space that we commonly use in everyday life is to operate within reachable space. So to pick up a coffee cup, for example, or reach up and get something down from a shelf. So things that are within reach of the arms and the legs. That's called egocentric space because it's referenced to your body. And that's the preserve of a part of the brain that's sort of at the top here, the parietal lobe. And this is part of the brain that is quite important clinically because there's a very common stroke pattern where one of the big arteries that goes up to that part of the brain will frequently become blocked, and people selectively lose the function of the parietal lobe. And a very weird thing happens to these people, which is that they lose a conception of part of the space around them. So what you can see here, on the left are examples where somebody who'd had a stroke is asked to copy the pictures. So on the far left are the pictures they were asked to copy, and then the next thing over is the drawing that they did. And then on the right, they were just asked to draw from memory some pictures. And you can see that this person who had a stroke on the right side of the brain has lost the ability to conceptualize the left side of space. So they don't draw the clock numbers on the left-hand side of the clock. They're missing the walls on the left-hand side of the house. They're not drawing the petals on the left-hand side of the flower, and so on. And even from memory, they are unable to... it's not that they can't remember that those things are there. They just don't really sort of realize that it's there. Interestingly, if you turned the clock upside down and now asked them to do those letters, they'd be able to fill them in. So they've lost... all of that stuff is in their brain. They just can't organize it. So that's the first system, reachable space. The second system is one that we use a lot, and it's the system that gets you around the world without really thinking about it. So when you commute to work in the morning, you're probably on autopilot for much of your journey. You drive down to the traffic lights, you turn left, you turn right. You know, sometimes you get to where you're going and you can't even remember getting yourself there. So these automatic kinds of behavior are the preserve of the striatum. So here it is, here that's it's lit up, it's the red inside this glass brain. And striatum is a very large part of the brain, and it organizes all sorts of things, not just navigation. It's this system that degenerates in Parkinson's disease, so it's probably quite familiar to you. And what seems to happen is that when you are doing some kind of spatial behavior, or indeed any kind of behavior, and you have to do the same thing over and over and over, the striatum picks up the problem and sort of says, don't worry about it, I know what we're doing now, leave it to me, you don't need to think about it anymore. So it takes on this habit, and it will just execute that habit, provided things don't change. However, if things suddenly change, so for example, let's say you're commuting to work and then you suddenly encounter roadworks, and suddenly you have to do something different, then now you have to reactivate the more thinking and less automatic parts of the brain and generate a plan. And that plan involves calling to mind a mental picture of the space that you're in, the layout of the streets or whatever, and you have to plan your way around that roadblock. So this mental representation of space is the preserve of the hippocampus. So the hippocampus is very deep within the brain, and we've known about it for a long time because we have known for a long time that it's important for memory. But a set of findings came along in the 1970s that made us start to realize that actually its role in memory is much broader than we understood up until that point. And in fact, it seems to involve making some kind of map of space onto which memories are attached. So it's a little bit like a memory palace, which is a concept you may have come across. So I'm going to come back to the hippocampus in a minute. I just want to tell you about one more system in the brain, which is not specifically involved with navigation per se, but contributes to this thing which I call spatial unease. And this is kind of a hypothesis at the moment rather than an accepted fact, but it's pretty well known that spaces have emotions associated with them. The brain is very quick to learn about the emotional valence of a place. So you go down a dark street and you have a bad experience, you'll never go down that dark street again. You might never go down any dark streets again. We're very, very quick to learn about spaces. I also think that we have this need to know about the space that we're in. And this is true not just for humans, but all sorts of other animals as well. So if you put a rat in an unfamiliar space, the very first thing it'll do is explore that space. And what it seems to be doing is trying to turn its sense of not knowing where it is into the sense that it knows where it is. It wants familiarity, and it's a very, very strong drive. And we have it too. Normally when we go to a new place, we kind of prowl around, we kind of open doors and we look, if you go to a hotel, for example, you would open all the doors, work out which is the bathroom, where's the fire escape, and so on. So we have this need to feel comfortable in a space, and that's probably, certainly the preserve of the emotional systems in the brain, which involve a number of structures, and it's hard for me to point them out, but one of them is the amygdala, which is down towards the bottom there. The amygdala is involved in a lot of different emotions, but particularly fear. And if people have the amygdala damaged, then they have these strange inabilities to be fearful of things that you and I would be fearful of. And the amygdala talks very closely with the hippocampus, which is the structure that we think is forming a mental map of space. So the anterior part of the hippocampus, the bit that's towards the front of the brain in humans, and the amygdala are right next to each other. And the anterior hippocampus has been associated with anxiety in humans and seems to be involved with processing threat of some kind. Now, how do we figure out how all this works? So we're trying to understand the language that neurons are using, as I mentioned. And in the 1970s, a really important discovery which was made by John O'Keefe at UCL, who was my postdoc supervisor, actually, in the 90s. And what he was doing was he had one of these microelectrodes implanted into the hippocampus of rats. He was trying to understand, actually, the role in memory. And he discovered, to his surprise, that if you take the nerve impulses produced by this cell. So those little spiky things are nerve impulses. He discovered that all of the nerve impulses from one of these neurons in hippocampus, you take the impulses and you look at where they occurred as the animal was walking around eating rice or whatever it was that it was doing. All of the action potentials, all of the nerve impulses would congregate in one part of the environment. So that cell would be very quiet as the rat was walking around, but every time it went into this one corner, this one place in the environment, it would suddenly become active. So O'Keefe called the place where these cells are active, he called that a place field, and he called these cells place cells. And he proposed, along with Lynn Nadell, a cognitive psychologist, that the hippocampus is the site of this mental map of space. And that has started several decades of work, all of which seems to support this idea. So it's turned out to be a very, very fundamental part of the brain. Now, in the decades that followed, people started to explore other regions of the brain that talk to the hippocampus, and they found a lot of other neurons that also seem to be involved in mental mapping. And one particularly important class of neurons is the head direction cells. So these are found in the green. That's a picture of the rat brain, actually. So you can see it's somewhat similar to the human brain, and the hippocampus is the blue structure. And in these regions around the hippocampus, they found neurons that become active whenever the animal faces in a particular direction. So they seem to be a little bit like a compass in the brain of some sort, presumably telling the place cell map which way around is which in the world that the animal is exploring. We know that the head direction cells don't use magnetism. They seem to use the familiar visual landscape that the animal can see in order to know which way around they should fire. And then finally, a very exciting discovery a decade or so ago now, from a team of researchers who were also trying to understand what information is being sent to the place cells. And they found in this region of the brain called entorhinal cortex that there are cells that produce place fields, very much like place cells do, but instead of just producing one, like a place cell would do, they produce lots of them. And most amazingly of all, they are very regularly spaced. They form this incredible polka dot pattern across the surface of the environment. So the black is the path that the rat walked as it was foraging around for rice, and the red is the little patches of activity of that cell. So the question is, how does the cell know where to put those blobs of activity? It seems to have to know how far the rat has walked as it goes from one region of the environment to the next, and also in what direction, because that's the way that it's able to line up those patches of activity in all of these neat rows. So this is a really remarkable thing to find in the brain. You know, the brain is usually a hopeless mess of signals going this way and that, and yet we've got this strange, almost crystalline structure of activity. So this is not a physical thing in the brain. This is a dynamic pattern that was laid down through space and time. It's really a remarkable discovery. And immediately suggests that maybe this is something like a grid reference for the place cell map or something like that. So O'Keefe, the discoverer of place cells, and the discoverers of the grid cells got the Nobel Prize in 2014 for those discoveries. Personally, I think the head direction cells should have also got the prize, because I think actually we've, in some senses, learned more from the head direction cells than anything else. For example, we've learned that the head direction cells need certain kinds of information in order to be able to set up their firing directions. And then if they can't do that properly, then all the other cells, the grid cells and the place cells, and you know, the basic sense of space doesn't work properly. So here's an example of where you might need that information. You're in an office, you know, one of these corridors with offices, very much like the one that I work in. You come out of one office, you go down the corridor and into the next office. How do you maintain a sense of direction across those spaces? You don't have a compass with you. Well, what happens is that when you walk out through the door, your brain tracks the movements that you made and updates your sense of direction as you go. So then when you turn and face down the corridor, your brain says, you've just turned, and it tells the head direction cells, you need to rotate your activity now because we just turned. And then you walk down the corridor and you turn again into the next door, and the brain says, we've just done another turn. Head direction cells, you need to update yourselves again because we've just turned again. And so then the head direction cells, when you go into the second office, are firing the same way as they were in the first office. They've managed to follow you around. And that's happening all the time. But for that to work, there needs to be some sense in which you've been able to move continuously from one space to the next. If you are just popped into a space, then suddenly the head direction cells don't know what to do because they haven't been able to track you. So for example, if you come out of a tube train, what are the head direction cells going to do? They have no idea how the space you're in links with any of the other spaces. Well, it turns out that what they do, if you don't give them any information, is that they guess. They say, well, we don't know which way around is which, but we will just fire this way around, and they'll pick an arbitrary direction. Now, if that direction doesn't align with the firing directions that they have in other spaces nearby, then forevermore you're going to have a slightly disconnected understanding of that fragment of space. And this is a very common experience in London. For example, you pop out of the underground at Covent Garden, and you pop out of the underground at Leicester Square, and you have different orientations in your head for these things because nobody told you which way round you were when you came out of the station. And then one day you walk from one to the other, and you go, God, I thought that north was over there, but it doesn't align. And as far as we know from studying head direction cells, it won't realign. You're always going to have a little bit of a mismatch in your map. So really, we need to be giving people information for their head direction systems wherever they go, if we're going to pop them into space in this very unnatural way. And then the final thing is that something that we've learned from the head direction cells is that rotational symmetry is really deadly for the head direction system. And rotational symmetry is actually very common in our architecture, not just circular spaces, but square spaces. Even a rectangular space has a twofold rotational symmetry. And the reason it's deadly is that the system is trying to use the visual landscape to help it orient. But if the visual landscape is ambiguous, then it immediately runs into difficulties, and it's having to rely on other things to take over instead, or conflicts will arise, and so on. On the other hand, mirror symmetry is no problem at all. So this is an example of a space that's mirror symmetric. So in that space, the view that you see as you turn around is unique. So there aren't any ambiguities in that space. It has pleasing symmetry for architects who like symmetry, but it's also something that the head direction system can work with. So these are some insights that we've developed from just the head direction cells, and we have many more that we could share with you. So the basic conclusion of my talk is that really we should talk more because we found out a lot in the last few years, and we would really like to get it out there, especially people like me who really love architecture and love the urban built space, would love to see some of these ideas make themselves known in architecture. And then to leave you just with that thought, I'm actually in the process of trying to collate everybody I can who's interested in this common problem of how humans navigate in space. So I'm trying to get together industry practitioners, engineers, cognitive neuroscientists, anybody who's interested in space. And I've created a special interest group in the Royal Institute of Navigation called CogNav, Cognition and Navigation. So if anybody is interested in being part of this community and joining the mailing list, then please let me know. I'd be really happy to add you. Thank you very much.